Good morning, everyone. As part of the future directions on emerging technologies related to bioequivalence assessment of topical drug products, in this presentation, I will focus on alternative and efficient BE approaches that are based on measuring the rate and extent of drugs bioavailability in the skin using sampling techniques such as dermal microperfusion and microdialysis. This is the standard disclaimer. And here are the learning objectives for my presentation. So I will start talking about different in vivo pharmacokinetics-based approaches that can be used to compare the bioavailability of topical drug products and have the potential use for bioequivalence assessment of this category of drug products. Next, I will present some of the advances that have been made through GDUFA-funded research related to developing and optimizing some of these cutaneous PK methods such as microperfusion and microdialysis and evaluate them as a regulatory methodology. The remaining portion of the presentation will cover how to design a bioequivalence study using in vivo cutaneous PK methods, what to consider during method development and optimization studies, and what to consider for the pivotal BE study. Measuring drugs rate and extent of bioavailability has been the most efficient BE approach for systemically administered drug products. During GDUFA, the agency has funded research related to cutaneous PK methods to support the development of more efficient pathways for approval of topical drug products that can be based on comparing their local bioavailability. These in vivo cutaneous PK methods include dermal open flow microperfusion or DOFM, dermal microdialysis or DMD, and epidermal or dermal pharmacokinetic tomography. So while the first two techniques will be covered in this presentation, the third technique is the focus of the following presentation by Dr. Ghosh. There are also other in vivo cutaneous PK methods such as tape stripping or dermatopharmacokinetics that is currently not the method of interest for BE assessment. DMD and DOFM are sampling techniques that can directly measure the in vivo rate and extent of drug bioavailability at or near site of action in the skin. In both techniques, we have a thin tube called also a probe that is inserted into the skin, passed through the dermis, and a physiologic solution is perfused into these probes and is collected from the other side. The drug that is released from the formulation partitioned into the skin and permeated into the dermis can diffuse from interstitial fluid into these probes, and by measuring the drug concentration in perfused sample, we can estimate the drug concentration in the dermis. And this has as a similar concept as when we take blood samples and measuring the drug in the blood samples or plasma samples. The difference between DMD and DOFM is that with microdialysis, the wall of these probes are made of a semi-permeable membrane, while in DOFM, as it's shown in this image, the wall of the probes have an open mesh design with half a millimeter openings, and the components of interstitial fluid can freely travel into the DOFM probes. Also here shows how 3 different microdialysis probes were inserted under each application site, and the samples are collected, and how you can use it to evaluate bioavailability of 3 different formulations simultaneously. So while DOFM is a relatively new technique, microdialysis has been around for the past 2-3 decades. And there have been traditional limitations and challenges associated with using these techniques to evaluate cutaneous bioavailability of drug products, and that is one of the main reasons that these techniques did not have regulatory applications. One of these limitations is the limited utility for certain classes of drugs, which is mostly applied to microdialysis because there we have a semi-permeable membrane and we have a restrictions for lipophilicity, protein binding, or molecular size of drug that may not be the case for the DOFM methodology. Also, one of the important challenges has been high variability in the data that was seen in the published studies using these techniques, especially microdialysis. And here is one example of a study where bioavailability of different metronidazole topical cream products were compared. This is not a GDUFA-funded research and was published in 2011. So the PK profiles of metronidazole from these products are matched. However, the variability in the data is very significant considering the log scale of y-axis, and the variability span over an order of magnitude. Also, one of the challenge is the dermal drug concentrations sometimes are too low to quantify, and you may need very sensitive bioanalytical technique to be able to measure drug concentrations in dermis. Another limitation is the mobilization of study participants while connected to large pumps and tubing during the study. That has shortened the study durations using these techniques. For example, most of the published studies with microdialysis were performed for a few hours, which is not enough to adequately compare the PK profiles of two topical products. And another challenge, which is an existing challenge, has been establishing acceptance criteria for BE and selecting the appropriate PK metrics for the comparison. So during GDUFA-1 and GDUFA-2, OGD has funded multiple collaborative research agreements related to novel methodologies such as DOFM and DMD to assess the bioequivalence of topical dermatological drug products based on the rate and extent of drug bioavailability in the skin. And these research agreements were awarded to UNM Research to evaluate the DOFM technique, and to Long Island University to evaluate microdialysis technique and fundamental cutaneous pharmacokinetics. At UNM Research, we studied the applicability of DOFM methodology as a tool for topical BE. There have been several clinical studies conducted to optimize and improve the precision of this methodology. Here are images showing how the location of the probes were marked on the skin, how the application template was used, how the probes were inserted into the skin, how the formulation was applied at each application site, how the samples were collected, how the application sites were controlled during the study, and how these small portable pumps were fixed to the subject and were used to perfuse the perfusate into DOFM probes. And having access to these portable pumps allowed the subject to be able to change position, or for example use bathroom, and therefore we were able to conduct studies up to 36 hours at UNM Research. One important aspect of designing clinical BE studies with DOFM at UNM Research has been to understand the sources of variability in the data and try to control and monitor them so that the method becomes accurate, sensitive, and reproducible, and can produce reliable results with precision. One of these study controls was application site that was controlled by using application template to make sure the distance between the application sites are consistent and distance among the probes are consistent. One important source of variability was the probe depth, which was monitored by taking ultrasound images of the application site at the end of the study and using them to measure the probe depth. Barrier properties of the skin can be different from subject to subject, and that can affect drug permeability to the skin. Therefore, barrier integrity tests such as measuring transepidermal water loss was performed in the beginning of the study, as it's shown here. Changes in the local blood flow can change the clearance rate of drug from the skin and can affect the concentration of the drug that is detected by DOFM probe. Therefore, the local blood flow was monitored throughout the study using an internal marker. Also, flow rate is another source of variability. Therefore, the flow rate of perfusate in the DOFM probe was monitored during the study, and as you can see, we observed minimal fluctuation in the flow rate. One important study that was conducted at UNM Research was a pivotal BE study for acyclovir cream product, where the US reference product was compared to itself and also was compared to a test product that was not expected to be bioequivalent to this reference product based on some in vitro studies. So here shows the cutaneous PK profile of a US reference product, and the method was reproducible and accurate enough to show that the US reference product is bioequivalent to itself based on both AUC and Cmax, and also was sensitive enough to differentiate the PK profile of the US reference product from an Austrian generic product, and showed that this product is not bioequivalent to the reference product. Also, if you look at the variability or the standard errors, you can see that the variability in the data here is very reasonable and relatively low, considering the fact that the data is not plotted on a log scale. Another study that was conducted at Ionium Research was to evaluate the bioavailability of EMLA cream and compare it to Oracix. So this was a pilot study done on a few subjects where EMLA, which is a lidocaine perlecaine topical cream, was compared with a dental gel product containing the same active ingredient, and the method was able to differentiate the PK profile of Oracix from EMLA for both lidocaine and perlecaine. At Long Island University, we evaluated the applicability of DMD technique for topical BE in preclinical studies. And here are the results of a preclinical study in rabbits to evaluate bioavailability of different metronidazole topical products, the reference gel, generic gel, reference cream, and the generic cream. And here are the cutaneous PK profiles measured by microdialysis. So the bluish colors are the PK profiles for the gel product that are very similar and are well differentiated from the PK profile of the cream products that are shown in red for the reference cream and the orange for the generic cream. And the variability in the data is also very reasonable, considering that the data is not plotted in log scale. So in the past decade, the agency has been attempting to develop efficient alternative BE approaches for topical drug products as a surrogate for comparative clinical endpoint studies. Efficient in vitro BE methods such as characterization-based approaches are particularly recommended for prospective generic products which have no difference in components, composition, or physical and structural characteristic of Q3 relative to the reference product. And Dr. Rani already explained this concept in the previous presentation. Now, the cutaneous PK-based approaches such as microdialysis or microperfusion that are efficient in vivo BE methods are particularly useful for prospective generic products which have similar components, composition, and Q3 compared to the reference product. In order to use DMD or DOFM techniques for demonstration of BE, the proposed methods need to be well-controlled, accurate, reproducible, and discriminative. All the study controls that I discussed in the earlier slide need to take into place to make sure the study is well-controlled and the results are reliable with precision. Method development and optimization studies are preliminary studies that need to be conducted prior to the pivotal study in order to show that the method is able to discriminate differences in the bioavailability and also to finalize and justify the method parameters for the pivotal study. These studies are usually done in a few number of subjects, so they don't need to be adequately powered. A pilot study can also be done prior to the pivotal study to compare the test and reference product. And the pivotal study is actually the bioequivalence study when you compare the bioequivalence of a test versus reference product and needs to be adequately powered. One of the studies that needs to be performed during method development stage is a dose sensitivity study to justify the selection of a dose for the pivotal BE study and to show that the method is able to discriminate differences in bioavailability among different doses. So in a study with EMLA cream, the target dose was selected at 10 milligram per centimeter square, and its bioavailability was compared to a lower dose and to the higher dose. So the DOFM method was able to differentiate the cutaneous PK profile of the 10 milligram per centimeter square dose from 15 and also from 5 milligram dose. It is very important to show that your method is able to discriminate the bioavailability of a higher dose than your target dose, because you want to show that the target dose is not selected as at such a high dose that the skin is at its bioavailability threshold and can limit the amount of drug that is going through it. Also, because if that's the case, then if a test product happens to deliver more drug than the reference product, your method may not be able to differentiate that. Also keep that in mind. Here we were able to show discrimination between a 10 and 15 milligram per centimeter square dose, but for a different drug product you may need to apply twice as much or 3 times more than your target dose to be able to show a discriminated PK profiles. And there may not be a linear or proportional correlation with the amount of dose you apply and the bioavailability of the product. Another study designed during the method development is to show that the proposed method is reproducible and is able to discriminate differences in bioavailability, and that would require including a positive control and a negative control for BE. So one example of a positive control is when you apply the reference product twice and compare its bioavailability to itself. So this was done for a acyclovir cream product where the reference product was compared to itself in a pilot study, and the red colors are showing the cutaneous profile, PK profile of Zavirax US cream product. And what's the negative control, you can use a different formulation of that drug. For example, in case of acyclovir cream, we used an Austrian generic product that had different bioavailability. So the method was able to differentiate the PK profile of that Austrian generic product from the US reference product. Another example of a study designed for negative control for VE is to use a different dosage form that contains the same active ingredient. For example, when our reference product was EMLA lidocaine perlecaine topical cream, we selected Oracix dental gel containing the same active ingredients at the same strength, and the bioavailability of the gel was compared with the EMLA, so the method was able to differentiate the PK profile of the cream product from the gel product. Another consideration would be to monitor the lateral diffusion and systemic crosstalk during the method development phase. So in order to study lateral diffusion, an untreated site or undosed sites can be located next to one of the application sites, and the samples can be collected from DMD or DOFM probes to evaluate whether we can detect any drug that, for example, is released from this formulation, absorbed in the skin, and diffused laterally, and so that we can pick up by these probes. And if we detect any drug in these probes, then we can evaluate whether there is any impact on our B assessment. Also, the drug that is absorbed in the skin can be picked up by dermal capillaries and get into systemic circulation and flow back and redistribute back in the skin. So it may have a confounding effect on the concentrations that we are measuring using these DOFM or DMD probes underneath the application site. In order to evaluate the effect of systemic redistribution, we can first measure systemic absorption of the drug by taking blood samples throughout the study, and also assigning an untreated site at the location that is far from the application sites, for example, an arm of the subject, and taking samples from the probes here and see whether we can detect any drugs and whether there is any impact on our B assessments. Another consideration for the study design is to finalize the dose duration and sampling duration for the pivotal B study. For example, in a study that we did with EMLA cream during the method development, here are the PK profiles we got for lidocaine and perlecaine. So the Tmax was observed around 3 hours, and we got a terminal phase. So based on these PK profiles, we decided to conduct the pivotal B study for 12 hours because by 12 hours we could capture most of the PK profile. However, if you have a PK profile like this in case of acyclovir cream, where you don't even get to Cmax during the study, then it would not be appropriate to conduct a pivotal BE study for 12 hours. Also, if you have a profile like this, one consideration would be use an alternative study design where you remove the formulation after a few hours, but continue sampling for the remaining duration of the study, and evaluate to see whether this alternative design can help to capture the Cmax. So after the method development and optimization studies, you need to finalize the parameters for the pivotal BE study. Also, for the pivotal BE study, you need to consider the number of test and reference sites per subject, the number of replicate probes underneath each application sites, and whether you want to use a larger application site and increase the number of replicate probes or not. Also, even though you have evaluated systemic redistribution during the method development stage, it is also recommended to include an undosed site on a different area and evaluate systemic redistribution during the pivotal BE study, because the skin permeation can be different from subject to subject. And as I mentioned, the duration of the study and the dose can be selected based on the results of the based on the results of method development study. So in conclusion, FDA is exploring cutaneous PK-based techniques to assess BE of topical drug products. Efficient in vivo DOFM or DMD methods have the potential to support demonstration of the BE when these proposed methods are optimized and controlled to be adequately discriminative and reproducible. If you have an efficient in vivo DOFM or DMD-based methods, then they can potentially be useful for generic products that have similar components, composition, and Q3 relative to the reference product. Also, the method development and optimization and pilot studies are needed to support that the DOFM study or the DMD study can differentiate changes or differences in the rate and extent of drug bioavailability in the dermis. And if you, as a generic applicant, wants to propose any of these techniques as an alternative BE approach for a topical drug product, you can submit a pre-ANDA product development meeting request to the Office of Generic Drugs. Here is the challenge question. Which of the following statements is not true about developing an in vivo DOFM or DMD PK-based method? The method development or optimization studies are expected to A, to show that an appropriate dose is selected for the pivotal BE study. B, the method development and optimization studies are expected to support that a proposed method is able to discriminate and increase in the rate and extent to which a product may deliver a drug into the skin. Or C, the method development studies are expected to be adequately powered. Or D, the method development studies are expected to support that an appropriate dose or study duration is selected for the pivotal BE study. And the answer is C, because as I mentioned during my presentation, the method development and optimization studies can be done in a few subjects and they are not expected to be adequately powered. So with that, I would like to thank and acknowledge my colleagues and supervisors at US FDA and our research collaborators at UNM Research and at Long Island University.